It is a great pleasure to be welcoming as our speaker Dr. Alison Adcock from Duke University. Dr. Adcock obtained her MD and PhD degrees from Yale University, which an MD, PhD takes not just a great deal of intellectual capacity, but a great deal of grit as well, to put it mildly. And she completed her medical training in psychiatry at the University of California, San Francisco. She joined the Duke Psychiatry and Behavioral Sciences Department in 2007, and she's currently Associate Professor of Psychiatry and Director of the Duke Center for Cognitive Neuroscience. She is very well known in our field for her research on how learning and memory are influenced by motivation and by motivational states, and we're going to hear quite a bit about that tonight, I believe. So please join me in welcoming Dr. Adcock.
Hello, you can hear me okay? All right. Louder. Okay. Just thumbs up at me if it needs to go up, okay? I'm going to try to bounce around to both sides of the room a little bit, but I am orienting to all of the slides on both sides, but I'm really honored to be here. And the Center for Vital Longevity has so many amazing colleagues that coming here and seeing you all together is really wonderful, and I'm just bowled over by the riches that the intellectual community and what you all have built here. Listening to you talk about a school for brain and behavioral sciences is really just, I'm dumbfounded at the idea that we would have a whole school for the things that we're talking about here this evening, so it's really quite something. So I'm delighted to be able to share with you all some of the work that we've been doing to understand the wonder that is the human brain. The brain is wider than the sky, Emily Dickinson wrote, for put them side by side and the one the other will contain and you besides. And I thought about this because of George talking about the wide sky last night when we were talking. And so I want to just put in front of you some images. I'm going to start by asking you to just experience them and observe the effect that they have on you. Observe what you think when you see them and how you feel. So how many of you suspected that these were golf courses when you first saw the very first partial image, the very first one, right? So did you recognize something in the shape of the space or the evidence of human art? What was it that clued you in? And I don't know if everybody in the room had the same experience. Probably some people were faster than others to think about this. But if you recognized it, did you have a little tingle of anticipation about what the rest of that field of view would look like? What else might be in the picture? Curiosity about what might have been outside? And when you were right, was there another little thrill of recognition? Right. So how about this one? Does it elicit the same or different feelings? Even though this one is a complete image, I imagine that those of you who are golfers are not only enjoying the remarkable beauty, but also feeling invigorated by the possibilities of what could happen there, right? And this sense of exhilaration and appetite has a chemical signature in your brain. Of course, that isn't the only way people feel when they play golf. And sometimes their motivations are less expansive and exploratory and more about just getting the dang ball in that tiny little cup And in fact, a golfer's focus may become so intent that they don't encode where they are relative to some very important landscape features, which may have consequences. Does anybody recognize this scene? This is Pebble Beach, and this is Jordan Spieth, who almost fell off the cliff trying to hit the ball. That's a 75-foot drop right there. And when he hit the ball, he ran in the other direction and apologized to his wife. So that's not the same little tingle, is it? That feeling of realizing that you're on a cliff. And so I ran through all of these. I hope, if it worked the way that I wanted, to have you experience some of the physical sensations that went with looking at a picture. Maybe activation, chills, or clenching, and some cognitive responses, questions or curiosity or a sense of expanding or contracting horizons. And all of this is to remind you that, well, not only, but to have you think about what those states are like and how often we experience them and don't really focus on them as something that we can train or use. So motivations are brain and body states, and motivation is not one thing, even though we have a singular word to say motivation. It's really like emotions, varied and related to the contexts, and you can think of them as emotions about possible outcomes that you have a role in making happen or not. So the motivational states that we're describing are also closely tied to the function of neuromodulatory neurotransmitters, neuromodulators like dopamine, norepinephrine, serotonin, that underlie these motivational states. And they create whole brain contexts in which learning happens. And we've discovered that imagination can really engage this neuromodulation so that you can configure your brain for learning or action or whatever's in front of you. So I use the word neuromodulator. What is that? A neuromodulator is a special kind of neurotransmitter, which are the chemicals that neurons release in the brain. And in classical neurotransmission, the neurons transfer information pretty directly one to another in a fairly specific way. So these tick marks represent the sort of ripple of activity, the action potential that runs down the end of a neuron into another neuron. And the target cells, their outgoing cells, integrate all the information that they get and send it on with another ripple of action potentials. And the effect of the first cells on the second cells depends on the kind of chemical that they're releasing, whether it's excitatory or inhibitory. So here we have an illustration of the purple neuron that's integrating the inputs from the red one, which is excitatory, and the blue one, which is inhibitory. So you can see a pause in the stream from the purple neuron, which makes it pauses firing for a little bit. So neuromodulation, unlike classical neurotransmitters that transmit information relatively precisely, modulators are more like fertilizer. They communicate a relatively uniform signal to a bunch of different neurons, a bunch of different places, and very a large increase in the number of neurons to the neurons that they're talking to. So it's dispersed. And they change the way these cells function and communicate. So dopamine is a good example of this kind of neurotransmission. Dopamine neurons in the midbrain receive inputs from widely dispersed brain regions about reward and other important things and distill those. This was a video, but I will have to be the video for you. So it distills, the whole cortical mantle has inputs to these regions, and there are very few cells. So you think about how many neurons there are in the brain, and these tiny, tiny clusters of neurons are distilling information from a huge number and transmitting that as a simple signal to the neurons that it talks to. So it's convergent and then divergent. So thinking about that kind of signal transform and what it means in the brain, It has really profound implications for brain function. Those broadcast signals, you can see that's the next slide. So the dopamine, serotonin, norepinephrine are all clustered in these tiny little nuclei that send their processes far and wide throughout the brain. And because of this dispersed impact, small changes in these groups of cells can have really big impacts on brain function. And that's why they're so often targets for drugs, for therapies, and why they're so important and implicated in illnesses like depression and schizophrenia, ADHD, and Parkinson's disease. So the association that most people have when they hear dopamine is reward. And we know that in humans, as in animals, the midbrain, where the dopamine neurons, the source of most of our dopamine, is activated when you're anticipating a reward. So this is an image of a functional MRI coded for activity with the colors. And you can see that there's a white outline around the ventral tegmental area, which is part of the midbrain, so it's the sort of most ventral part, and flanked by the substantia nigra, which is the most people know about because of its association with Parkinson's. So the ventral tegmental area is more affective and emotional and communicates with cortex, and the substantia nigra communicates more with the striatum and motor function. So these two small parts of a small thing we can segregate them both functionally and with behavior. And the disease states that attack the dopamine neurons in Parkinson's disease spare the ventral tegmental area, so it remains intact for longer as people suffer. But there's a lot more to it than that, as you might guess from what I've been saying. So for one, activity in these neurons elicits exploratory behavior, like sniffing, curiosity. Animals and people become more active in the presence of dopamine release. So this looks a lot like curiosity, but there are we stuck? Next slide. Yes, okay. And this effect is the one that got me thinking about our ability to regulate dopamine. So this is a very technical slide, but it's really just a record of one neuron talking to another neuron. So even though that was a very low-level recording of cells talking to each other, the thing that, the observation that's interesting is that in order to enhance the communication from one cell to another, that communication can be enhanced if dopamine is present in the synapse, in the prep, before you start the stimulation. So this is not dopamine that's released by cells firing. It's not released by an external event. It's there to begin with. So the idea that there would be something there anticipatory that would facilitate plasticity was the very low-level question that got me thinking about the psychological implications and what we could use them for. So you didn't need the slide for that. But despite all the different things that dopamine does, it doesn't go everywhere in the brain, like norepinephrine, which is really uniformly distributed. It goes to special places. It goes to places that are really closely, Okay. These are parts of the brain that are really important to learning. So not sensory cortex so much, not motor cortex so much, but the places where connections need to be made. So, and in particular, I really wish you could see this slide. So to a region named by early anatomists for its resemblance to a seahorse. Does anybody know what this is? I know there are people in here that know what this is. So the hippocampus, and if you could see the slide, you would see an actual seahorse and then the dissected human hippocampus, which really does look like a seahorse. And is it going? Oh, well, we unplugged this, though. Where did? Yep, I'm going to put that back in. Okay. Is it going to happen? Okay, well, I'm not waiting. So the seahorse-shaped hippocampus, as we have known for decades, is really important to memory function. And we typically associate it with things that people call memories, autobiographical memory. But it's actually important to lots of processes in the brain that require making connections between multiple things, placing them in relationship to each other, and then using that information to extrapolate. So problems that you've never solved before, ideas that are new combinations of old memories, all of these things clearly depend on hippocampal function. In addition, knowing where you are and knowing what behaviors are appropriate, like where you can smoke and where you can swear and where you should not hit a golf ball, like all of that sort of mapping of your environment is critically dependent on this system, which is the seahorse hippocampus covered by cortex that feeds into it. Now, I'm just paging through my bullet points to get back to. Okay, so based on the function of this system, the neuronal plasticity data that I showed you earlier for a brief moment, and what we know about the way dopamine is triggered to be released in the brain, which is by anticipation of action that's required, and more reliably elicited with rewards, we started to investigate whether anticipation of reward would enhance memory formation. So the very first way that we did this was in a crass, dumb, paying people to remember things paradigm. So this seems quite obvious, but oddly enough, it had never been tested. So we had a monetary version of the "will this be on the test" question that we get from students all the time. So we would give a heads up for each thing that we showed a student. These were mostly Stanford students who, the first study, am I in your way? Who participated in the first study. So we gave a heads up for how much the next thing would be worth if they recognized it on a test. And so we had really high dollar items that were worth $5, and we had cheap items that were worth 10 cents. And as you might expect, the $5 items were better remembered. And the 10-cent items are still, there's still memory for these things, except for the one smart aleck who closed his eyes for all the 10-cent items because he knew about psychology and interference. But mostly, there's a distribution of reward and relationship to reward. And the important thing, though, is that none of this is given during learning. So there's a reward cue, and there's a promise for it, but they came back for the test the next day. So everything that was happening was about the anticipation. And we broke it down into the period between the reward cue that we gave and the picture that they were supposed to memorize. And that was the interval that we were interested in, what was happening in the brain. So if we go and look in the brain using functional magnetic resonance imaging, which allows us to isolate things pretty precisely, then we see that the same brain regions, the ventral tegmental area, that are activated by reward anticipation, hitting buttons to earn money, predicted whether the upcoming item would be remembered. And that relationship swamped the things that were happening while the picture was being processed. So this brain area where the dopamine neurons live was active while it was waiting for the thing that was to be memorized. And that activity predicted memory for the upcoming item. It was quite robust. And it was there, the relationship is there for the low value items also, but really impressive for the high value items. So in addition to that, just activation in the midbrain where the dopamine neurons are, there was a connectivity between the midbrain and the hippocampus. So thinking about what the effect that the midbrain is having on the hippocampus is something that's been occupying my lab for the last 15 years. But the more clinically relevant, actionable effects are thinking about the variation that we saw. So there was enough variation from trial to trial. So not every time you saw $5 did your brain react to it. And that allowed us to see the relationship with memory. And also across individuals, there was a really strong correlation between your average activation to reward cues and your performance on the high value items. It was one of the strongest, cleanest correlations I've ever seen. And sadly, there were people who were down in the basement. So they looked like they were almost deactivated after the reward cues, and they did not do better on the high value items. They were actually, if anything, inhibited. So this question of what was happening with the people who were not responding to the rewards the way we expected them to, and not benefiting from the memory boost that they should have got, and how we broke that for them, is another big focus of the lab right now. So I had a cute slide with a golf ball right in the middle of that question. But so the fact that it's breakable, that we can interfere with this reward benefit in many different ways, including if you have a grumpy graduate student who's testing the participant, right? So, and we did have this. We had somebody who just couldn't get this to work, and then I asked what they did when they walked into the student, to test people, and it was clear why they weren't responding to rewards. But there are lots of ways. That variability is a question for us to answer. But we had experiments to do, and we wanted it to work all the time. So instead of trying to guess what we would use to drive the midbrain, we started thinking about, well, what would happen if you just asked people to activate this region? What if you just told them, this is a region that we think is related to motivation, and so if you imagine being motivated, will your midbrain be active? So who thinks they could do this? Yeah. That's Duke students, right? So I thought that people would just be able to instantiate, simulate that state, and be able to drive activity without a reward cue. Because what's the reward? So it's a 5, right? It just says 5. It's not even an image of $1. So we scanned participants. We put them in the scanner, and this big metal tube, and they're lying there, and we say, okay, imagine being motivated, what does that mean for you? Really simulate it. And the answer was no, they could not. So they did not reliably drive activation in this brain region. So then and the way that we triggered this was with just a white square to say go. When they saw the white square, they would imagine, and it didn't work. So then we gave them a thermometer that was a gauge of the activation in the midbrain. And we instructed them to try out different strategies so they weren't just hammering on the same one. And we asked them to tell us what they were thinking about. And so after about 20 minutes of training, we tried again to give them a white square and say, activate your midbrain, and they did it. So after this short period of sampling different imagery techniques and looking at what worked, when we asked them to apply what worked, they were able to sustain dopamine regions activity for a period of about 20 seconds each time, and when we brought them back later, they were able to continue to do this. So this was quite surprising to many people who study these parts of the brain, who have very, I think, well rationalized and reasonable idea that this is beyond the reach of cognition or ideas, that these regions are almost reptilian and they're so basic that this kind of cognition shouldn't really affect them. They're really low-level information processing. So we had to do a lot to convince people that this was true. But it's now replicated multiple times with multiple control groups, and we have populations so we have been doing the self-activation in attention deficit disorder and showed that after a session of about a half an hour and then another 30 minutes later, we have impact on effortful task performance that is very similar to what happens when people take stimulants. So the task that gets used is, here's something that's really boring, but you'll get paid really well for it. Can you keep doing it? And the rational decision is, if the chances are high that I'm going to get it, then I'll work. And if they're low, then I won't. And if they're high, then I won't, because it doesn't matter. So it's really a very tailored effect that supports the impact of this activation on dopamine-sensitive behaviors. We have colleagues who are testing this in depression, and the results are not as impressive because the folks who are the depressed patients that they were working with were not as able to generate the activation. And when we look at the kinds of things that they say they were doing, for our first study, we have very endearing kinds of strategies that people report to us. You can imagine family members doing well, running marathons. My favorite one was actually one of our students in the center who said, I just imagine running down a line where thousands of people are giving me high fives. So, and the depressed patients are using words like work and job and, compulsion, right? Things that are shoulds or musts. So we have to figure out how to help people who are stuck in the shoulds and the musts to access those same kinds of little thrills that maybe you experienced looking at the golf courses earlier. Okay, so there are a bunch of plots, which, oh my goodness, that video is working, Nick. It's so sad. So there is a video showing, we were trying very hard to get it to display, and I had despaired. But the other interesting thing that happens during this learning to self-activate, which we have begun calling cognitive neurostimulation, is that the interaction of the midbrain changes over the course of learning. So while people are processing feedback, there's activation that is correlated between the midbrain and the striatum, which is brain region that is associated with feedback-based learning. And then as we take the feedback away and ask people to reproduce their mental imagery, then the connectivity between the midbrain shifts to the hippocampus. So this sort of structure for simulation comes online to help people generate activity in the midbrain. So for basic science, this is really exciting. And for clinical applications, we have ready ones that we're trying and expanding all the time. But there's still this question of when the rewards don't work the way that we expect them to. And we haven't tested these self-activation paradigms in as many different behavioral situations as we have for extrinsic incentives, where you promise people money or something fun that they enjoy. But in addition to the variation in VTA activation, we're starting to get hints about what might be going on with people who are not benefiting from the rewards. So when we want to test hippocampal function, one way that we do it is have people navigate in mazes of some sort, usually virtual mazes, and look at how they learn where things are. So they're learning where rewards are, they're learning to stay away from things that will be penalties, and they map out a space. So in these environments, people's behavior is much richer, and we have a lot of different elements that we can look at to see how they are stitching together their representation of the environment. But one of the striking things that emerges from these kinds of paradigms where people are learning and responding to reward cues and looking at or sometimes punishment cues, is that the impact varies across time and individuals in a very specific way. So when we test the impact of punishment on this kind of learning, if it's hippocampally dependent, map making, it routinely, reliably impairs performance to be working to avoid a punishment. So that is a very robust phenomenon, and we can measure it with skin conductance. So the kind of lie detector and sweat and chlorine channels that are that probably go along with getting pins and needles. If we look at the way that people's skin responds to a reward cue, for the people who are getting reward cues and responding with that kind of arousal, they look very similar to the people who are working to avoid punishments. So in other words, they are responding to the reward cues as though they are threatening. So this, we have more and more evidence now that relates this peripheral signal to activation of the amygdala and the function of the neurotransmitter dopamine and its source, its tiny source, in the locus ceruleus. So we have a picture emerging where it's not enough to activate midbrain, you also have to have this other system damped. And we have evidence for this in a few different paradigms and a few different ways of looking at brain activity. But over and over again, the pattern is not that there's competition between the midbrain releasing dopamine and the amygdala norepinephrine system directly. It's that that other system is inhibiting the effect of the dopamine on the hippocampus. So they're tracking together, things that are salient and important are tracking together, but this norepinephrine system is preventing this interaction from happening. So we can think of a few different reasons why this might occur, but the mental memory representations that emerge from learning in these two states are also important to think about. So some of the images that, I'll see if I can describe the state of, well, actually we have the golf ball getting to the whole image and the concentration and focus that occurs when you're limiting and narrowing your focus is highly related to norepinephrine function. And the amygdala seems to generate a brain state that shunts the detection of important stimuli to the cortex overlying the hippocampus in order to create these simple representations that will drive behavior. So if you think about what you need to know to get a golf ball in a hole, it's very limited and sparse, and the area around is irrelevant at best and distracting at worst, right? So those kinds of cartoon representations, the one that I often use when I'm not thinking about golf is how do you represent a bus? So if it's big and yellow and moving at you, that's really all you need to know to not get hit. So simplifying those features of big and moving at you is not a default state or it didn't happen that you generated a lot of extra information and memory. It's actually tailoring the representation to create an efficient communication to your motor system. So in contrast, if you have to take apart an engine and put it back together, that kind of focus on pieces without putting the whole together is not useful. And in that case, we want the norepinephrine system to be released and let the midbrain and hippocampus talk to each other to generate this rich tapestry of treasure map. So these two systems have contexts in which they're really important and function to keep us safe and moving through the world. And for most of us in modern life, the utility of the sparse representations that drive an automatic rapid response is overrepresented. We have lots of things to do with our brains that require us to make these really complex representations. And maybe that wasn't true in evolution. Maybe we had more tigers to run away from. Whatever the case, it's really perversely hard to get away from that system that's making things simple so that you can act quickly. And we see the evidence of that in just how easy it is to break these reward benefits, right? We can offer people incentives that in some cases work, and as soon as we shift them into perfectionism or threat by somebody who's in the room with them or even a frown on somebody's face, that reward doesn't have the effect on the brain that we might want it to. So it's not enough to just dangle extrinsic incentives. It's not enough to just pay people to do things without thinking about how the brain is responding. So I think I would love to be able to tell you a little bit more about the specific anatomy and the way that it's set up to support these functions, but I don't think I can curl myself into hippocampus and show you all the subregions. But I hope it's clear that I'm not saying that there's one state that we should all be in all the time. That sort of rigid idea that there's, a positive thinking that will help us make our way through every situation is not the idea. It's really thinking about what is the current situation and how you map your motivational states onto what needs to get done. And given that it's so easy for us to have that tunnel vision and harder to generate the distributed map of all of the things that are important to you and keep them active, I think that is the one that we will be mostly charged with trying to help each other learn to do. And the advantage, though, is that instead of just telling people not to think of things or suppress the information, it's cognitively a lot easier. It's not as much of a magic trick to shift from negative to positive. It's just letting people know that if you activate a whole system of your goals and incentives in your current awareness and try to keep them present, that you will have a response that permits fluidity. So instead of trying to tell people not to think negative, think positive, just representing the whole spectrum of things that you care about as you're moving through the world, even when it's really important to perform. Seems like a strategy that we can use will be easier than a lot of things that we try to teach people to do. So I'm excited about this way of thinking about motivations and how we regulate them, and just developing a broader vocabulary that is maybe as rich as our vocabulary for emotions, as we think about what are the brain states that will help us do the work that we need to do and connect and take care of the people that we care about. So one of the easiest ways to think about it is just the facility for pretending, right? Imagining that you are a superhero or Marie Curie or somebody. And the developmental literature is full of examples of things that the properties and capacities that kids adopt when they implement this whole image or schema of what behavior is. And there was a really nice study from one of my Duke colleagues recently that showed that girls that were pretending to be Marie Curie persisted in solving problems longer and had much more, okay, well that's just in time for the current slide, which is my daughters with the trappings of industry and scientific inquiry at the kitchen table. Maybe it'll come up, but even if it doesn't, the awareness that they had of what it looks like to be a scientist, or even just sit with glasses and pretend to be reading a paper when you're not old enough to know how to read, but you know that reading glasses are important, right? So, okay, so I want to, tame these pretending skills that we have when we were younger, and think about what kind of motivations we can support in each other going forward. So if I have one take-home message, it is hope is medicine, and hope is not the same as an imperative goal that you have to get done. It is sitting with the uncertainty that exists around you and thinking about how to incorporate it into an informed and realistic action plan, which sounds complex, but we all know how to do it pretty instinctively. So I have a little time for questions, and I would love to hear what you all have to say. Thank you.
Thanks very much, Alison. So we don't have to imagine that you're a superhero. I can't. I'm sure your arousal level is. If we had a thermometer here, it would be at the top.
Yep,
So
there's a lot
thank you so much for such a cogent talk under such difficult circumstances. And we have time for questions. And I'm seeing someone here who wishes to ask.
I want to hear about your test subjects. And did you have any people that were deaf, people that were blind, people that lacked senses to stimulate themselves to think differently?
No, we have not.
Hold on.
Yeah.
Could you repeat
I
the
will
question?
repeat the question. So the question was, what happens when people have changes in their sensory capacities that prevent them from having the kind of visual imagery that I used to illustrate these things with you all? And we have not done it, but I think there are a lot of people that study populations that you describe, deaf, blind, and show that their representation of the world is really rich. So all of the sort of reward anticipation and where are things that matter to me should be completely intact. And so they would have, and I think that actually the specific scenarios that people are describing, the images that they generate, are not what we're teaching people. So they're not rehearsing the same images over again. I think we're teaching them to tune in to that feeling that I was trying to elicit with the images at the beginning of its emotion. Some of it may be physical, but there's also a change in the way that you think, when, a change in the way that your brain is attuning to things that they should have access to. So these systems don't depend on.
Question here.
Sonia. Oh.
Hi. I'm a fellow Duke colleague as well, but as a child psychiatrist, I think one of the greatest things that I have come to experience is that in working with children, adolescents, and adults, to generate some of that dopamine when they come in quite in despair with depression, is to identify heroes. And I loved your idea to encourage that, because it does make an impact emotionally, physically as well. And I wonder, have there been any studies on the impact that we now use all the emotional support animals and pets? Because now when you go into Duke Library and other libraries during test time, we bring in the emotional support animals, and the students light up, and they relax, and they take a break, and then they can return to their work. And one of the most encouraging experiences I've had is when one of the neuroscientists that I went to a lecture for, he made a point of saying a joke every 5 to 10 minutes. And he got the audience laughing, and it was interesting. He was testing our memory in between to generate that. So I think there's loads of interesting things. Have they done any work with the animals and pets? I think that there are certainly people who are clinically working with animals and pets, but I don't know of anybody who systematically looks at those interactions and their impact on cognition. But as you say, just that relaxation and backing off from something that you're really stuck on. I think there are lots of ways to accomplish these changes. Exercise is probably one. The feeling that your body can move and you can do things with it, and sometimes it surprises you. I mean, those are all experiences that I think would contribute to changes in your motivational and neuromodulatory status. So,
I'm going to take a question from this side of the room.
Yep.
Just
where did I see? Okay. Next there. I'm going to.
So we're trying to alternate.
Yes, thank you.
Sorry.
Have you looked into putting into practice this with PTSD? Because thinking of those the triggers.
Yes. So I think that's a really exciting approach. And we know that hippocampal function is important to PTSD, and that intact hippocampal function is predictive of fewer symptoms. So the idea that you could engage hippocampal function in a wide variety of, I gave you the list of things that it does, but I think PTSD, if you think about it as not sufficiently localizing a threat, right, that it's everywhere. Yeah.
Desensitizing to the threat or whatever, something
So,
like that.
well, the desensitizing is one approach, and people definitely try this. But there's also, I think what I'm suggesting is that there could be a way that localizing it and knowing where it is more precisely could be also helpful. And that, I think, is the approach that this kind of engaging the hippocampus as you reactivate the memories might be helpful for.
Yes, please.
Thank you.
Duke, class of '84.
Hello.
Back when dinosaurs roamed the earth. I'm a local therapist, and I work with people with PTSD using EMDR. I don't know if you're familiar with that. But I lead an artist way group and very interested in play and creativity. And so this is just pinging all of those areas. And I'm wondering, have you looked at, with fMRI, anybody doing EMDR? We do a very simple, imagine your best friend who makes you feel nurtured. Now let's expand that, and then we tap into that brain state later to help someone, to give them a resource for processing traumatic memories. Have you looked at that at all?
No, I think I don't do EMDR research, but I know that there are people investigating it. I don't know of mechanistic research. Do you know of the eye movement desensitization research? Do you know?
Yeah, I know of the eye movement desensitization approach, but I don't know of any studies that have looked at the neurobiological basis of it, particularly with the hippocampus.
Yeah.
But
Or any. So I have big questions about how it works. Okay.
So hi, Alison, Mustafa Hussain, another Duke colleague of Alison. Thank you very much. I mean, it was an amazing talk, and especially without slides, the way you did it, I would have crashed along with the computer. So my question is, the title of your talk was Memory and Motivation. And while you're talking about memory and cognition, dementia or Alzheimer's disease comes to your mind.
Of course. Yeah.
So are there any studies looking at motivation for that purpose, encouraging people that can help in terms of retaining memory? Thank you.
I'm not aware of studies, Kendra, but there are certainly people thinking about motivation and its impact on cognition as people age. And at North Carolina Central, NC State, Tom Hess has a study that I cite a lot that I think about, that is looking at the typical pattern of slower reading and comprehension in older participants relative to college students, that was completely reversed when he gave them a passage to read that actually mattered to them. So when the content was not, are you going to the student center to pick up a kayak, but it was, you're going to get your driver's license, and you got, then they were just as fast and just as accurate at extracting information. So I think there are two interesting ideas. One is that the efficiency of our brains as we age and acquire more information and more understanding of the world and more patterns that go in is, some of what we're testing with a lot of these paradigms is a different mode of interacting with the environment, right? And the other is that if we're enhancing motivation, we should expect to see memories improve, cognition improve. So the idea that this encouraging yourself to feel curious, encouraging yourself to feel engaged, and putting yourself in positions where you experience novelty, all of those things should help all of us.
We're going to take one last question.
Okay. Yeah.
Thank you so much for the stimulating talk, and thank you
My pleasure.
for reminding me what a magnificent part of the country you've been able to do your work in, since I am a very grateful graduate of the University of North Carolina.
Yep.
I'm glad to see that it's not only the Tar Heels that are doing good work.
No, we try, yeah.
I'm curious about the, since your original images that you were portraying to us, I assume was to stimulate the kinds of reactions as you progressed through certain types of images and so forth, until you were not by your fault, but you were literally cut off. That it occurred to me to ask, how long a period of time was your research with live specimens and their responses, and the collection of all of that data and so forth? How long a period of time, as represented by those images we were looking at, were your subjects exposed to?
So you're asking within a particular study, within one study?
Yes.
Yes.
And if you know how many studies we were talking about with those images related to. The reason I'm asking is because they were all linked to golfing.
That's right.
One way or another.
Correct.
There were no golfers, with one or two exceptions. So we had no idea of what level of play we were talking about, or possibly, or
Right.
who the golfers might have been,
Right.
what the tournaments were.
Right.
And the period of time, if I were a researcher in the lab, I think I probably would have been a suicide, because over the last 2 years, the world of golf has been turned literally upside down by the Saudi Arabian leadership and the organization of an entirely separate tour. And I don't know if you follow the sport or not, or read
I am not a golfer. Yeah. So I think the thing about, so you're not done. Yeah.
No,
Yeah.
the question is, was the period of time over which you did your stuff, collected your data, and talked to us about it,
Yeah.
might have been affected,
Yeah.
particularly with the areas of the brain that we're focusing on
Right.
in your reporting of your work?
Yeah.
Do you think it had any effect?
Well, I do think it would have an effect, but the images that I chose of golf courses were partly because golf courses, I think, are a nice natural stimulus of a landscape that, and I didn't use them for my studies. I used them for you all here because I think it's easier to connect to those images than the ones that we use for research, which are much more boring. And it's a video game of a big circular pool that you're swimming around, right? So those were for the talk, not for the science. But I think your question of what the meaning attached to those, whether it would be loss or instead, some poignant homesickness, right? So we have lots of associations with images that are, I was assuming that almost nobody here would know the specific courses that I was pointing to. I certainly don't, except for the Pebble Beach one, because it was so striking. But those really gorgeous ones, I have no idea where they are. They are just landscapes that have threats and rewards embedded in them and have a particular association for people that use them. Yeah. So thank you for that very interesting question.
Yep. So, yeah. It's interesting. So essentially, I think, Jack, you're in a sense restating, I think, what Alison was saying is that not everything, it's not one size fits all for these stimuli and for what gets people in the right state. So it's been an interesting evening. I would like I know we've already acknowledged your heroic efforts under very difficult circumstances that are completely out of your control. You'll be getting the bill for the broken projector later. But I think your talk was fabulous. We actually did not need the slides to understand the central messages of what you were saying, and particularly the extremely important take-home message. So I'd like everyone to join me again in thanking Alison for a tremendous talk. We are scheduled for another public lecture in April, at the end of April. This will be on the main campus. Hopefully everyone in this room will be getting a heads up about this in the fairly near future. And we'd love to see as many of you as can make it at our next public event, which I believe is April the something. Say again? 19th. April the 19th, I'm told reliably. So we hope to see you all there. Thank you all for coming tonight. I hope you all enjoyed the presentation. I apologize, although it's not my fault, that the audio visuals were not up to scratch. But as I said, I don't think that detracted from the talk in any significant way. Thank you all very much, and good night and drive safely.